I'm going to be speaking about hydroxychloroquine. Hydroxychloroquine is a therapy that's very interesting because it was approved in the 1950s, a very long time ago, for the treatment of rheumatoid arthritis and lupus. And I bet some of you might be wondering why we are investigating such a therapy in the context of the coronavirus crisis. Next slide, please. Well, decisions are being made quickly and rightly so in the current situation. There are a number of therapies that are being trialed for the treatment of coronavirus, and our clinical colleagues have the intuition that these anti-rheumatic drugs could be useful when things get out of control and patients enter what they are calling a cytokine storm. And there's a need to control that storm and to calm down the immune system. There's a few examples of therapies that we are investigating, including more advanced and novel treatments like IL-6 inhibitors like tocilizumab that has been approved by the Chinese regulators. But you have probably heard that hydroxychloroquine has now been approved by the FDA also for the treatment of coronavirus. And this was announced by the President of the United States recently. Next slide, please. So the truth is that we don't have good quality data on the efficacy of hydroxychloroquine to treat coronavirus. And we will have to wait a little bit further until we have such data from observational data sets in Odyssey. But what we do have is a lot of retrospective data on the use of hydroxychloroquine for other purposes and for other intentions. Next slide, please. So we have focused on conducting a coastal estimation or population level estimation study looking at the safety profile of hydroxychloroquine. We designed a comparative cohort analysis comparing uses of hydroxychloroquine to uses of sulfasalazine, which is another similar therapy for rheumatoid arthritis. And we also designed a self-controlled case series, which is a study design where patients are compared to themselves to minimize confounding. And in this second case, we also wanted to look at the safety profile of this therapy regardless of indications and not restricting to the rheumatoid arthritis population. Therefore, the participants of the first study are people with rheumatoid arthritis who are new users of hydroxychloroquine or sulfasalazine, while in the second, this is going to be users of hydroxychloroquine who then at some point have one of the outcomes of interest. That's why it's called case for self-control case series. Next slide, please. The outcomes that we're investigating include severe adverse events, including arrhythmia, cardiovascular disease, venous thromboembolism, liver failure, kidney failure, GI bleeds, and mortality. And the second bit of outcomes that we are now reanalyzing include flu, viral infections, and hospitalized pneumonia, which we will also be looking at in the future in the comparative cohort, but not in the self-control case series. Analysis-wise, data analysis-wise, we are using propensity scores to minimize confounding based on observed variables. We are using propensity score stratification and negative control outcome calibration to minimize the impact of any potential residual confounding. And we are conducting an on-treatment and an intention to treat analysis. In the SCCS analysis, we are adjusting for age and season because these are obviously factors that change over time and therefore are not adjusted for by design in the self-control case series. Next slide, please. So this is the largest study to date on the safety of hydroxychloroquine. It's a study that has so far been run in 4 databases, and there are more to come. But so far, we have accurate data on more than 130,000 users of hydroxychloroquine compared to 57,000 users of sulfasalazine. And for this particular event, venous thromboembolism, we also had a lot of people having that event of interest, as you can see. Next slide. Before we looked at the results from each of the analyses, we looked at the diagnostics of the models to make sure that these were robust enough to go ahead finalize the analysis. We looked at the overlap in the propensity score distributions, as you can see to the top left of this slide. We looked at how the propensity scores managed to reduce confounding in the observed variables, as you can see to the bottom left of the slide. And finally, we also looked at negative control outcomes, which reassuringly suggested that there isn't any residual confounding left after the propensity score analysis. Next slide. I'm not going to go through each of the individual outcomes because there's plenty of them, but I will invite you to go to these websites and basically play and have a look at the different outcomes that were investigated. This is just an example of venous thromboembolism, again, where you can see how there is no association between the use of hydroxychloroquine and the incidence or the risk of venous thromboembolic events. Next slide. This is just a zoom in so that you can see more closely the same results with hazard ratios both without and after calibration that are very close to the null effect. Next slide. So, in summary, we have completed the biggest study to date on the safety of hydroxychloroquine. Reassuringly, we are not finding any consistent signals, safety signals, that make us think that this drug is not safe, and we are therefore thinking that it is quite a safe medication. We have some work in progress. We are running across the whole network more analysis, and we will be accumulating more data on this association between hydroxychloroquine and the different safety outcomes. And we are also waiting the final results from the self-control case series, which I hope to have in the coming couple of days. And finally, outstanding and waiting to be implemented, we are interested to learn the antiviral efficacy of these treatments, and we will conduct a new user design amongst COVID-19 infectees when data starts accumulating and growing for us to be able to do that.